Good morning, everyone. I'd like to thank you for giving me the opportunity to present my research to you today. So, crop disease is a very important issue in the world today. Just to give you one example, and this isn't the pathogen that we're studying in particular, but just to give you an example, UG99 fungus, also known as black stem rust, is a pathogen which can potentially attack 80% of the crops in developing nations today. And it has the potential to spread across continents in just a matter of months, affecting the food supply of up to 1 billion people in Africa and the Middle East alone. And if you can imagine, this is just one single pathogen out of the thousands that it could potentially affect crops all across the world. So as scientists, our job is to first attempt to understand the mechanisms by which these pathogens attack the crops, and also to understand the defense mechanisms that the plants have in place to attempt to protect themselves against these pathogens. And in our lab, we use Arabidopsis thaliana as a model organism to understand the defense pathways of plants. So Arabidopsis thaliana is a model organism for several reasons, one of which is its relatively small size, making it easy to work with, as well as its small genome, which has been completely sequenced. Further, it has a germination to seed production period of only 5 to 6 weeks, as well as it is also easily transformed, meaning that we can easily introduce new DNA into the plant. Also, several mutants, or actually rather, there's mutants available for pretty much every gene in Arabidopsis' genome. So we never, as scientists, we never have to worry about making new mutants. We know that they're already available for us if we choose to select them to work with. And finally, the great thing about Arabidopsis is that its defense pathways are analogous to the defense pathways of other crops, such as rice as wheat. It's just that Arabidopsis is much easier to work with, making it really an ideal organism to work with. So in Arabidopsis, we already have a decent idea of the defense pathways in place. We know that salicylic acid is the key defense molecule in Arabidopsis. And further, we know that that SID2 is a gene that is involved in the biosynthesis of salicylic acid, and that ACD6, ALD1, EDS1, PAD4, and EDS5 are genes involved in the accumulation of salicylic acid. And NPR1 is involved in salicylic acid signaling, in defense signaling rather, downstream of salicylic acid. Now in our lab, we have a unique mutant, ACD61, which is a very powerful tool in understanding Arabidopsis defense. So ACD61 is a gain-of-function allele, and as you can see, ACD61 compared to wild type is much smaller in size. And it also has greatly elevated levels of defense, as well as very high salicylic acid levels compared with wild type Arabidopsis. And as you can see, well, I don't know if you can see that well, but it has some like yellow leaves compared to the wild type. And those are actually sites of spontaneous cell death, which is a hallmark of constitutive defense in Arabidopsis. And finally, ACD6, as I mentioned, is a very potent tool in studying defense. Now the reason for that is because ACD6's dwarfism is actually defense dependent. In other words, if we take any defense gene and we knock it out in an ACD6 plant, the plant actually becomes larger in size as compared with ACD6. Not quite as large as wild type plants, but somewhere in between ACD6 and wild type plants. And this is a very powerful tool because, well, for two reasons. First of all, we can use ACD6 to identify just visually new defense genes. In other words, if we mutate if we mutate various genes, we can see which ones cause ACD6 to be larger. and the mutations that cause ACD6 to be larger, we can speculate are defense genes. And also, ACD61 can actually be used to dissect defense pathways in Arabidopsis. Now, the way it can be used to dissect defense pathways is, assume that we have two defense genes, gene X and gene Y in Arabidopsis. If gene X and gene Y are in the same pathway, then knocking out either gene X or gene Y should have the same effect on defense, right? Because you're basically inhibiting the same pathway in either case. And knocking out both genes at the same time should also have the same effect because it's still all in the same pathway. Therefore, remembering that ACD6's size is defense dependent, knocking out both genes at the same time should produce an ACD6 plant which is roughly equivalent in size to knocking out either gene. Now, let's assume that gene X and gene Y are in different pathways. Well, then in that case, if we knock out both genes, then the overall effect on defense should be greater than knocking out just one of the genes, right? So therefore, we would speculate that the mutant plant that has both genes knocked out in ACD61 should be larger in size than just knocking out one gene or the other. So basically, using this hypothesis, the question we attempt to answer is, are PAD4 and EDS1 in the same pathway? Now, the reason we ask this question is because, and as I mentioned, PAD4 and EDS1 are both involved in salicylic acid accumulation, because previous work has shown that PAD4 and EDS1 interact physically at the molecular level. So this gives us good reason to speculate that, well, they might be involved in, they might be both in the same pathway if they interact molecularly. Therefore, to answer this question of whether they're in the same pathway, we compared several different mutant plants. First of all, just ACD6 by itself, then ACD61 with just PAD4 knocked out, ACD61 with just EDS1 knocked out, and then ACD61 with both genes knocked out. And as kind of a comparison, to provide a good contrast to this, we also compared EDS1 with SID2 knocked out. Now, the reason we did that was because we know that EDS1 and SID2 do not interact molecularly, so that would kind of provide a good comparison for us. And here are the results we got when we grew these plants in the same flats. So as you can see, ACD61 by itself, it has the highest levels of defense, and accordingly, it is the smallest of all the plants. Now, if we compare the ACD6 EDS1 PAD4 triple mutant, we can see that it is roughly the same size as the plants in which we just knocked out EDS1 and we just knocked out PAD4. But if you look all the way on the right, we can see that the plants in which we knocked out both EDS1 and SID2, well, you can see that is larger than the plants in which we just knocked out SID2 and EDS1. And this is the data basically quantitated. And I did a statistical analysis on this data, and I found that the triple mutant in which EDS1 and SID2 was knocked out was statistically larger, statistically significantly larger than the plants in which just SID2 and EDS1 was knocked out. And furthermore, I found that the plants in which just EDS1 and PAD4 was knocked out was not statistically significantly larger than the single, well, rather the double PAD4 and EDS1 mutants. So in conclusion, we determined that EDS1 and PAD4 are in the same pathway. And basically what we did was we provided genetic evidence to support the studies that have shown that these two molecules interact at the molecular level. So we've basically supplemented the previous knowledge in the field. And furthermore, we've determined that EDS1 and SID2 are potentially in different pathways. And this also genetically supports the fact that they do not interact molecularly. So for future directions, basically, we've provided some proof of concept work here in which using ACD6, we can really dissect the defense networks in Arabidopsis. And actually we've, just like the work I presented for you today, we've actually already isolated and have planted several different triple mutants of all, basically all of these defense genes in Arabidopsis. And we are in the process of analyzing this data, and this will hopefully kind of clear up, or rather elucidate the exact pathways by which these defense genes work, and hopefully, ultimately, this work will apply to other important crops in the world as well. All right, thank you very much. Are there any questions? Oh, right. Yeah, of course, I would like to thank my mentor, Dr. Hua Lu. It is. I've been in her lab for 3.5 years now. It's been a wonderful experience for me. Also, I'd like to thank Tim Lee Swift and Guying Wang, who both work in the lab as well. And of course, I'd like to thank Mark Ustar, the Meyerhoff, and the undergraduate research awards programs at UMBC. Okay, thank you. Are there any questions?
Yeah, you say, I think the results pretty show that EDS1 and PAD4 are definitely the same pathway, but you said that your surmising they interact, but wouldn't you get the same results if EDS1 and PAD4 were the same pathway but separated by different steps in the pathway? Or, you can't really determine the order of the pathway, can you?
Right, so we can't, you're right, we can't determine the exact order of the pathway.
Because you just said they were interacting, so do you mean like directly interacting or just surmising that?
Well, we do know from previous work that they do actually directly interact physically. Now, whether they are in like subsequent order in a pathway or whether they're further separated, we can speculate that they're directly like next to each other because of the fact that they physically interact. But our work basically just confirms that they are in the same pathway. We can't speculate further based on our work alone. Any other questions?
In a sort of a general sense, that I'm surprised that the act of defending yourself would make you smaller. So in general, do the plants defend themselves by killing off infected parts of the plant? Is that why they're small?
Yeah, that's actually a hallmark of, it's called systemic defense. Basically, I guess the plant's logic is if you kill parts of yourself, then bacteria can't grow there, right? That's probably, I mean, that's probably what's happening, and that's pretty much what stunts their growth as well.
Yeah. So cutting off your arm if you have an infection.
That's exactly the analogy I was using earlier. Yeah.
It can grow back. That's good.
Yeah, in the case of plants, it can grow back. Right? In our case, it can't.
All right. Thank you, Sasan.
Okay. Thank you very much.